Hi, I'm Olivia, and I'm presenting on the introduction and case studies of Chapter 24, where we discuss infections of the central nervous system. From this chapter, there are a couple things that you should be able to take away. The first chapter objective is correlating the anatomy and physiology of the nervous system with the infectious processes that are affecting them. You will also be able to describe symptoms that can help differentiate nervous system infections caused by viruses from those caused by bacteria and parasites. You will also be able to relate pathogenic mechanisms to disease prevention strategies for microbes that infect the nervous system. And the last chapter objective is being able to correlate the physiology of each pathogen with its antimicrobial treatment. So, just for a brief introduction, the first section of this chapter has to do with a case study of a male by the name of Miguel. Miguel was very sick and had an unknown infection. The first part of this chapter introduces rabies. Rabies is one of the most deadliest infectious diseases known to humans. Animals usually spread this disease through their saliva. Once the disease is in the human body, rabies attack the central nervous system and usually cause death. The case study of Miguel describes that he is a 19-year-old man from Mexico. He worked at a sugarcane plantation in Louisiana. One day, Miguel felt extremely tired, his left shoulder hurt, and his left hand was numb. Over the next 3 days, his conditions worsened and he was taken to a hospital in New Orleans. Miguel experienced many signs and symptoms previous and during the infection. So he experienced fatigue, pain, numbness in his left extremities. He developed a fever of 38 degrees Celsius or 101.1 degrees Fahrenheit. He had trouble breathing, unresponsive to external stimuli, fixed and dilated pupils, bradycardia, and hypothermia. So bradycardia is a slowed heart rate, and hypothermia is low body temperature. And on the pictures in this slide, you can see that I have a picture of eyes and then a picture of a heart scan. So in the picture of the eyes, it shows undilated and dilated pupils. In Miguel's case, he had dilated pupils, so his eyes looked more like the right side of the picture, where the black part of the eye is way bigger than normal. And then in the picture of the heart scan, you can see someone who has a normal heart rate and someone who has bradycardia. So in the normal heart rate, the scan is pretty evenly spread out with the lines being everywhere, and there's bigger and smaller bumps. And then in the bradycardia, there's less lines and there's more spread out because the heart rate is slowed. While Miguel was in the ICU unit, which is the intensive care unit, he had a lumbar puncture done, which is another word for a spinal tap, and this showed his cerebral spinal fluid from his lumbar puncture. And in this, they found the rabies infection that he had. This ended up being a very specific virus, IgG and IgM, which is in his CSF and his sternum. So, along with this, his subsequent EEG showed severe cortical impairment, and this basically means that his reactions were slowed and he's more confused and less reactive. Obviously, this was due to the infection of the rabies that had gotten into his brain. So he had to get extubated, and then he died shortly after from the invasions of the virus.
Hello, my name is Taylor Student, and I will be covering section 24.1, which is the anatomy of the central nervous system. The section objectives are to describe the functional organization of the central nervous system, explain how meninges and cerebrospinal fluid protect the central nervous system, describe the differences between two major types of neurons, and correlate the anatomy of the central nervous system to the symptoms when it is infected. The central nervous system is the control center of the body. It has two main parts, which are the central nervous system and the peripheral nervous system. The CNS controls and coordinates everything, while the PNS collects data from the periphery and outside environment to communicate the information to the CNS. They consist of specialized cells called neurons. The sensory neurons send information from the sensors in the periphery to the CNS. The motor neurons send information from the CNS to the periphery. Each neuron has a cell body and specialized extensions called axons and dendrites. Information is transferred between axons and dendrites through the cell body. Some axons are covered with myelin, which protect the axon and accelerate information transfer. This image is a comprehensive visual overview of the nervous system and highlights its key components, functions, and associated diseases. So let's break it down into sections. The central nervous system includes the brain and spinal cord. The peripheral nervous system is composed of cranial nerves, spinal nerves, and the ganglia. The cranial nerves emerge from the brain and serve areas of the head and neck. The spinal nerves are divided into regions, which are 8 cervical nerves, 12 thoracic nerves, 5 lumbar nerves, and 5 sacral nerves. The chain of vertebral ganglia is a part of the autonomic nervous system, controlling involuntary actions like heartbeat and digestion. The CNS is protected from most harm by several mechanisms. The skull and vertebrae are physical barriers to trauma from the outside environment. This figure also includes a microscopic image of neurons, showing the complexity of their connections and structures. This figure illustrates the meninges, which are 3 thin membranes that separate, cover, and protect your brain and spinal cord. The meninges include the dura mater, which is the outer layer closest to your skull, the arachnoid mater, which is the middle layer, and the pia mater, which is the innermost layer closest to your brain tissue. These membranes provide a support system for blood vessels, nerves, lymphatics, and the cerebrospinal fluid that surrounds the CNS. The pia mater secretes a clear fluid derived from the blood called cerebral fluid into the subarachnoid space. The subarachnoid space is between your arachnoid mater and pia mater. It is filled with cerebrospinal fluid, which cushions and protects your brain and spinal cord. We produce approximately 500 to 700 milliliters of CSF every day. Elevation of the CSF pressure is very serious and can cause damage to the brain and spinal cord. This figure shows the parts of the vertebrae. The picture to the left shows the superior view of the vertebrae, and the one to the right shows the left lateral view. We have 33 vertebrae stacked from our neck to our tailbone. The Spinous process is a bony projection on the back of each vertebrae that serves as an attachment point for muscles and ligaments. The Spinous process's primary function is to provide support and movement to the spine. The intervertebral discs allow the vertebral column to be flexible and act as shock absorbers during everyday activities such as walking, running, and jumping. The spinal cord is separated from the vertebrae by the spinal meninges. A pair of spinal nerves exit the vertebrae from each side and provide nerve supply to the structures around the vertebrae. These images represent a lumbar puncture, also known as a spinal tap. The lumbar puncture is performed when a person is suspected of having a central nervous system infection. When a person has a CNS infection, a sample of their cerebral fluid will be removed, which allows doctors to analyze it in a lab so they can diagnose the condition. The fluid is tested for red and white blood cells, protein, and glucose. The clarity and color of the fluid are also checked, and it is tested to see whether bacteria, viruses, or abnormal cells are present. The lumbar puncture can diagnose infections such as encephalitis, meningitis, and Guillain-Barré syndrome. During this procedure, a spinal tap is inserted between the third and fourth lumbar vertebrae into the subarachnoid space using an aseptic technique. This is normally performed under anesthesia and takes around 15 to 30 minutes.
Hi, my name is Morgan Harris, and I'm going to be covering section 24.2, which is an overview of infectious diseases of the central nervous system. The learning objectives for this section include being able to categorize CNS infections by the anatomical structures affected, being able to use key laboratory information to distinguish types of CNS infections, and being able to compare and contrast the roles of meninges and cerebrospinal fluid in the pathogenesis of CNS infections. An infection of the brain or central nervous system must be diagnosed quickly and correctly to avoid severe disease and possible death. This is hard to do sometimes, and CNS infections are often misdiagnosed due to vague signs and symptoms at the beginning of the infection. Most CNS infections begin with flu-like symptoms and often cause headaches, dizziness, and fatigue, which are all common symptoms of less serious illnesses. Damage to the CNS can range from mild to severe. Usually, in most cases, symptoms gradually become more serious. This was presented in the opening case study. Signs and symptoms of infectious diseases of the CNS do not typically point to a specific offending agent. Healthcare providers must identify other changes in blood or cerebrospinal fluid to make accurate diagnoses. The biggest obstacle in treating CNS infections is the blood-to-brain barrier, which blocks not only pathogens but also antimicrobial agents. To reach the CNS, microorganisms use various paths which depend on the invading agent. Most microbes first infect distal sites, or sites that are further away from the CNS, and then travel to it. Other ways pathogens reach it are through infection in an adjacent bone or through a structure closely associated with the CNS. These structures can include the eyes, sinuses, ears, or the pharynx. There are a few organisms that can target the CNS directly. For example, some mosquito-borne viruses enter the circulation because of a bite and travel straight to the CNS. The same goes for tick-borne viruses. CNS infections are generally rare, but are almost always caused by viruses. Infection of the viruses are often caused after humans cross paths with their reservoirs. This can include ticks, birds, or other humans. Depending on the virus, transmission can also happen after inhaling contaminated droplets, eating or drinking contaminated foods or liquids, or through direct contact. There are 3 major diseases associated with CNS infections. Those are meningitis, encephalitis, and myelitis. Meningitis is inflammation of the meninges and develops when the meninges are infected by a microbe or irritated by a chemical or biochemical. Some symptoms include fever, severe headache, and confusion. Encephalitis is inflammation of the brain and also results from an infection or a reaction to a chemical or biochemical. Encephalitis can be a primary or secondary disease. Primary encephalitis is the result of a direct attack on the brain. Secondary encephalitis is caused when the infection or inflammation spreads from the meninges to the brain. Encephalitis is often caused by viruses and are transmitted through insect bites. The main difference between the presenting signs and symptoms of meningitis and encephalitis is that encephalitis will cause disruption in a patient's cognitive, motor, and sensory functions, and meningitis will not. This image shows the difference between a healthy and normal brain and one that is infected by encephalitis. As shown by the arrows, the small clusters of white are the encephalitis infection. Viruses can also cause myelitis, which is inflammation of the spinal cord, or radiculitis, inflammation of the spinal nerve root, where the spinal nerve exits the vertebra. Meningoencephalitis is a combination of terms used to diagnose inflammation of the meninges and brain. The combination of terms is used because more than one site is infected. That can be done through infection or by chemical insult. Inflammation of the brain may lead to cerebral edema, which is swelling and excessive fluid retention in the brain. This produces an increase in intracranial pressure, causing severe headaches. It can also cause fever, fatigue, nausea, vomiting, and neurological changes. These neurological changes can include paresthesia, which is numbness, tingling, or a pinprick sensation, hyperesthesia, an extreme sensitivity, and auriflexia, which is the absence of reflexes. Changes in mental status, memory, and temper are common, as well as confusion, disorientation, and irritability. Diagnosis often requires examination of cerebrospinal fluid, or CSF, for chemical and biochemical composition and the presence of ineffective agents. CSF from a healthy person is usually clear, containing no cells. Pleocytosis is the increased number of white blood cells in CSF, which is often found in patients with meningitis. The type of white blood cells found in the CSF may offer a clue to the cause of infection. Finding polymorphonuclear leukocytes in the CSF indicates a bacterial infection, and the presence of lymphocytes typically indicates a viral infection. CSF is best obtained before starting antibacterial treatment so that killing the bacterial pathogens before the laboratory can grow them can be avoided. A spinal tap is often used to acquire CSF. The CSF is then tested for glucose and protein concentration and for white and red blood cell count. To the right is a chart that shows normal, viral infection, bacterial infection, and fungal infection CSF specimens and includes the number of white blood cells, glucose, and protein in each of these. Treatment of CNS viral infections is aimed to reduce inflammation of the brain and intracranial pressure. Some antiviral agents are used to treat selected viral encephalitides, but there are no medications currently that can treat viral cases. Bacterial, fungal, and parasitic infections of the CNS are aggressively treated with antimicrobial agents in combination with supportive measures. Even with quick diagnosis and treatment, death and permanent damage are big threats to patients. When it comes to prevention, vaccination is the best means. Vaccines are only available for a minority of viruses and bacteria that affect the CNS and are not available in preventing parasitic or fungal pathogens. Due to this, preventative measures are largely aimed to avoid contact between us and potential infectious agents. This is done through reducing insect populations by fumigation, venturing outdoors only at certain times to minimize insect exposures, and reducing contact with infected people and animals helps reduce incidence of infection. State and local health departments are helping monitor outbreaks and make them public knowledge. Controlling mosquito populations by means other than fumigation is also being researched. For example, having infected mosquitoes sent to attack other infected mosquitoes will help prevent the spread of disease.
Hi, my name is Mackenzie Brooks, and I am going to be discussing the Chapter 24.3, Viral Infections of the Central Nervous System. To start off, we're going to look at what the different kinds of viral CNS infections are. So meningitis and encephalitis are the two major categories of viral CNS infections. CNS infections, which are caused by viruses, can be extremely dangerous to the patient. Fortunately, serious viral infections of the CNS are uncommon. They typically resolve favorably in patients with a healthy immune system. Most viruses that attack the central nervous system tend to infect the meninges and cause aseptic meningitis, which means there is no bacteria present. Some other viruses that attack the CNS prefer the brain tissue instead of meninges and produce symptoms of encephalitis. Now, what is viral encephalitis? This can be described as a rare disease that causes the brain to swell, as seen in the image over to the right. On the left, you have the normal brain, and then on the right, you can see the areas where it is shown to be swelling. Viral encephalitis can be categorized as either primary or secondary. with rabies falling under the primary category. Secondary encephalitis is caused by meninges caused by herpes simplex virus type 1, childhood diseases like measles or rubella, and autoimmune reactions followed by viral infections. One viral infection of the central nervous system is called IMPACT, otherwise known as Bell's palsy. And what is Bell's palsy? It is a weakness or paralysis of the muscles on one side of the face. Some viral infections can result in inflammation of cranial nerve 7, the facial nerve. The inflammation damages the nerve so that it can no longer relay messages from the brain to the muscles of the face. So when the affected person wants to smile, but only his eyes are the things moving. Bell's palsy is a condition that affects nearly 40,000 people every year in the United States, but unfortunately, the cause is usually unknown. The next viral infection of the central nervous system is rabies. This is one of the deadliest of all infections known to humans, but it is rarely seen in the United States. Rabies virus is endemic in different mammalian animal populations, including fox and most commonly known, bats. This is an RNA virus, which is bullet-shaped, as you can see in the image to the right. It can be described as neurotropic, which means it prefers to replicate in the nerves. This also comes in 3 different forms: encephalitic, which makes up 80%, paralytic, and atypical. Some treatments are immunoglobulin and rabies vaccines, but if they are administered before the symptoms begin. Once the patient begins showing symptoms, it is very unlikely that you will be able to cure them. And going back to the mode of transportation, silver-haired bats are the main mode and the most commonly known. On to the right, you can see a depiction of the silver-haired bat, which usually infects humans and other animals. Some symptoms of rabies, they begin usually vague, including headache, fever, or nausea. Extreme symptoms usually begin after 2 to 3 months, including neurological symptoms. The main ones include hydrophobia, which is the fear of water, aerophobia, which is the fear of fresh air, aggression and confusion, quadriparesis, which is weakness of all 4 limbs, and this can even progress to a coma or death if the case is severe enough. Rabies. The incubation period is usually 2 weeks to 12 months. Several tests are conducted, but the main one is quantitative reverse transcription polymerase chain reaction, otherwise known as QRT-CR. During this test, they look in patients' saliva, serum, spinal fluid, skin, and hair follicles to find the rabies antibodies. This can usually be confirmed post-mortem when the Negri bodies are seen in brain neurons. Even though rabies virus is a very known virus, it can often be misdiagnosed, especially when looking in organ donors, which unfortunately ends up transferring it to the next patient who is trying to receive those organs. And if you look over to the right, you can see that the nucleus and the cell membrane are surrounded by the Negri bodies, which is the telltale of rabies.
Hello, I am Marissa Renato, and I will be talking about viral encephalitis. Mosquito-borne encephalitis types are Eastern equine encephalitis, EEE, and West Nile encephalitis, WNV. And within the Eastern equine encephalitis, it is transmitted from bird to bird by mosquitoes, and then it can be passed to horses and humans from these mosquitoes. It has a 5 to 10 day incubation period, and there is no vaccine. but that it is extremely rare and has a high fatality rate of 35%. And it is made of a single strand RNA genome. And within this, there was a case story. In this story, there was a vet student named Rich. He was very tired and then he had developed a fever. A few days later, his father had noticed he had developed confusion and he had developed no appetite. So they had brought him into the hospital. They had asked for his patient history. With him being a veterinarian student and being close to the water, they had an idea of what it may be. And they had ran tests. By this point, he was paralyzed. And then this eventually led to his death. Then to continue with the West Nile encephalitis, the WNV, it was discovered in Uganda and the first case was shown in New York in the United States. It is transmitted in the bird population, and when it infects patients, it usually does not develop disease. It can cause minor symptoms such as headache or vomit, but it's like flu-like symptoms in this case, and it is usually way less severe. It is a little less rare, but it is not usually deadly. I feel this picture better describes how the EEE is contracted in humans. To start, the EE virus is transmitted from infected animals to birds where they amplify, meaning the mosquito, which is the vector in the situation, takes its nutrients from an infected bird, and then from there, it can lead to the second step where mosquitoes carry the virus from birds to dead-end hosts, such as horses and humans. The only way an EE virus can be acquired is via a bite of a mosquito that has been fed an infected bird. So this means there is no other possible way for a human or a horse to get this virus. So it has to be done through a direct bite from a mosquito. That's also a part in the factor of what makes it so rare. And I feel this picture clearly shows how people contract it. Now to continue, I'll be talking about viral meningitis. To start, viral meningitis is not as severe as bacterial meningitis. It's known as aseptic meningitis as CSF fluid is clear. Usually this just ranges from headaches to nausea. It has a 7 to 10-day incubation period, and there is vaccinations for this, which are most of the time received in childhood years. Then to continue with a more specific type, lymphocytic choriomeningitis virus, LCMV. It is a enveloped single-strand negative RNA virus, and it can cause aseptic meningitis. It causes elevation in the lymphocytes in the CSF. The household mouse is usually what can contract this, and this is if you inhale such as their saliva, urine, or feces, or this can be contracted from mother to fetus, and there is no effective antiviral. For poliomyelitis, it is very contagious, and it's an enterovirus. It can be transmitted through fecal-oral route or directly from person to person through their saliva, and then it can multiply in the throat or the GI tract. It has a 3 to 35-day incubation period, and it can affect all parts of the CNS system, or known as the central nervous system. Most infected patients remain asymptomatic. Only 1% of symptomatic patients develop paralysis. And an example of this would be the woman who had spent 60 years living with a iron lung almost, and living off an iron machine because she would die without it until she had eventually did pass. And then vaccine efforts have come close to eradicating this disease almost fully, through the Salk vaccine and the Sabin vaccine.
Hi, my name is Isabella DeBonis, and I'm going to be covering bacterial meningitis. So, meningitis is considered a medical emergency, and some of the symptoms include fever, stiffness of the neck, and sudden onset of severe headache. There are 3 different types of bacteria that can lead to further health issues with meningitis. So, one of them is Neisseria meningitidis, which can cause sepsis down the road. And complications are a major reason that antimicrobial treatment fails for meningitis, which is why vaccination is really essential against all 3 of these bacterial pathogens, and it has majorly reduced the rates of the deadly disease. However, Listeria does not have a vaccine since it is a new infection. So for the case study, there was a 4-month-old from Saudi Arabia who was hospitalized with fever, tender neck, and a purplish spots, and upon hospitalization, the clinician was suspecting meningitis, so they took a CSF sample, and a gram stain examination revealed that there was gram-negative diplococci inside his white blood cells. The child was treated with antibiotics, and he did make a full recovery, but when his father brought him in to the hospital, he was clinically well, but along with the father and the patient's 2-year-old brother, they both tested positive for N. meningitis in their throat cultures. The father and all of his children were infected with the same strain. First, I'm going to be talking about meningococcal meningitis, which is a bacterial infection of the brain and spinal cord that is caused by Neisseria meningitidis. This can be spread through direct person-to-person contact or through contact with contaminated fomites with saliva or mucus. Meningococcal meningitis uses specialized villi to adhere to and invade epithelial cells that produces an endotoxin and causes those purple rashes that you see in the picture on the right. This can also cause bloodstream infections, which is why vaccination in all preteens and teens is essential. Next, I'm going to be talking about listeriosis. Listeriosis is when a person becomes infected with Listeria bacteria when they eat foods that the bacteria has infected. It is a foodborne pathogen, and it can cause fever and diarrhea. It is a rare but serious infection that can be treated with fluids to prevent dehydration and antibiotics for invasive illnesses. If it's an intestinal illness, most people can recover without treatment. The CDC estimates that each year, 1,600 people are infected and 260 people die each year of listeriosis. Complications like meningitis and brain abscess usually occur in pregnant women and immunocompromised individuals. Infection during pregnancy often leads to fetal infection and death. Lastly, there is neonatal meningitis that infects newborns. This is a very serious, life-threatening infection that causes inflammation of the meninges, which is the protective membranes around the brain and spinal cord. This usually occurs within the first 28 days of life. That's why premature babies are at a higher risk because their immune systems are less developed, which makes them more vulnerable to this infection. Bacteria from the mother's vaginal secretions are a main source of infection. The most common sources of this infection are E. coli and Group B streptococcus. Pregnant women who test positive for Group B streptococcus receive antibiotics during labor to prevent neonatal meningitis transmission to their babies. If infected, those infants will most likely have neurological issues, which can cause likely vomiting, diarrhea, and difficulty breathing.
Following up after the bacterial infections of the central nervous system, my name is McKenna Haggerty, and I'll be starting to discuss the section of bacterial neurotoxins. Starting off with the case study, Mitch, a 47-year-old resident of Oklahoma, was admitted to the hospital with rapid onset of progressive dizziness, blurred vision, slurred speech, difficulty swallowing, and nausea. Findings on the examination included drooping eyelids, facial paralysis, impaired gag reflex, and no fever. He developed breathing difficulties and required mechanical ventilation. The patient reported that during the 24 hours before onset of symptoms, he had eaten an at-home canned green bean and a stew containing roast beef and potatoes. Analysis of the patient's stool detected botulinum type A toxin, but no Clostridium botulinum organisms were found. Mitch was hospitalized for 49 days, with 42 of those days being on mechanical ventilation before getting discharged. Botulism is a disease that causes complete loss of muscle function. It is one of the most potent known toxins that affect our nervous system. Botulism is a food-borne bacterial neurotoxin that is caused by an obligate anaerobe, the gram-positive spore-forming bacillus called Clostridium botulinum. Using secreted exotoxins, 2 microbes cause lethal paralytic diseases. Voluntary muscle functions fail to respond to the mind because the botulinum toxin interferes with neutral transmission. In botulism, phylactic paralysis arises from the lack of excitatory neurotransmitters. Symptoms of botulism typically appear 12 to 36 hours after ingestion. Infants that consume Clostridium botulinum spores often from the environmental exposure or honey. After the spores begin to grow in the infant's stomach, they release the toxin. After the toxin is released, muscle weakness and paralysis will appear in the face and spread to other parts of the body. The second neurotoxin I will be discussing is tetanus. Tetanus is when C. tetani spores enter the body after trauma is endured. The most common cause of tetanus is rusty metal. This acute infectious disease is caused by the spores of Bacterium Clostridium tetani. Symptoms typically begin 3 to 21 days after infection. Initial signs include muscle stiffness near the wound and difficulty swallowing. Some symptoms that come with tetanus are intense muscle contraction, muscle spasms, and locked jaw. Necrotic tissue creates an environment without oxygen, which is needed for germination. As the bacteria grows, they release tetanospasmin, a toxin that enters the nerve cells near the injury site. The toxin travels up the nerves to the spinal cord, where it attaches to certain motor neurons that normally help control muscle movement. These neurons release chemicals that help slow down nerve signals. The tetanus toxin blocks the release of these chemicals, allowing nerve signals to become too strong and frequent, causing muscle spasms seen in tetanus. Tetanospasm to muscles to contract. The image shown is a photomicrograph of Clostridium tetani, and the image is painted by Charles Bell, showing a patient with muscle twitches from tetanus. Similarities between tetanus and botulism are 30 to 40%. Each of the toxins have similar structures and almost identical modes of action. Each toxin is initially composed of a single peptide that is cleaved after secretion to form a heavy and light chain. Each chain remains secured to each other by a disulfide bond. Heavy chains include a domain that binds to a ganglioside on the nerve cell membrane and a translocation domain that makes a pore in the nerve cell. The toxin or light chain passes through the pores and into the cell. The light chain disrupts the intracellular movement of exocytic vesicles containing neurotransmitters needed for muscles to relax or contract. The photo on the left demonstrates the basic structure of tetanus and botulinum toxins after cleavage. The first step is binding domains to receptor molecules or gangliosides of the nerve cell. It is followed by the translocation domain making a pore for the passage of the toxin. Finally, the protease toxin disrupts the release of the neurotransmitter. The photo on the left is mechanisms of action botulinum toxin. First, the neuromuscular junction inset shows vesicles filled with neurotransmitters. Toxins then bind to the heavy chain and the toxin is endocytosized into the nerve terminal. Finally, the small subunit of the toxin enters the cytoplasm, cleaves the target, and makes the nerve terminal unable to release acetylcholine. This slide depicts the tetanus toxin that enters through the nervous system at the neuromuscular junction. Once tetanus enters the It then travels up the axon to an inhibitory neuron of the central nervous system. When it reaches the neuron, the toxin divides proteins that are associated with exocytosis of vesicles containing inhibitory neurotransmitters. When there is an absence of inhibitory signals, excitatory signals from acetylcholine prevail, stimulating muscle contraction as shown in the picture. Both of the diseases can be prevented. There is no vaccine for botulism, so following proper cooking procedures is important to avoid it. However, there is a tetanus vaccine given during childhood. Although it does not provide lifelong immunity, people who are exposed can get a booster shot. Antibiotics are not used to treat either disease because the bacteria does not cause the illness directly. The main treatment for both diseases involves antitoxin injections and supportive care for the patient. If botulism goes untreated for an extended period of time, there are risks of severe, irreversible complications. For example, respiratory.
Hi, my name is Ava Brunetti, and today I will be covering Chapter 24, Section 5, Fungal Infections of the Central Nervous System. Fungal infections, also called mycoses, usually begin due to another infection from another area of the body that disseminate through the bloodstream to other areas. These can be incredibly lethal, and a majority of the patients that are affected are immunocompromised. Many of these fungal infections target the elderly, recent neurological surgical patients, and AIDS patients. The use of steroids or certain medications can also increase a patient's risk. For example, if spinal steroidal injections are contaminated, a fungus can slip through the host's immune system and begin an infection. This was seen in 2012 when a group of patients were given a spinal steroid shot from a contaminated batch of steroid medications. Due to this direct injection in a neurological procedure, the contaminated pharmaceutical was able to pass right through the host's innate immune system and cause an infection, also known as meningitis. As mentioned in the last slide, one fungal infection of the central nervous system is fungal meningitis. This is a very broad term because it can be caused by several different types of fungi. There are a broad range of signs and symptoms, all of which include a fever, headache, stiff neck, nausea, vomiting, sensitivity to light, and confusion. Like I said, they can be caused by many different types of fungi, which include Candida albicans, Coccidioides immitis, Coccidioides posidazzi, Cryptococcus neoformans, and Cryptococcus gaudy. The first form of fungal meningitis is Candida meningitis, which is caused by Candida albicans, which is most commonly known for causing yeast infections. This is the most common cause of fungal meningitis, and it can also cause brain abscesses as well. Candida is a natural inhabitant of the skin, oral, vaginal, and gastrointestinal biota. The dissemination through the blood to visceral organs is what causes an infection. Candida is a dimorphic yeast, which means its pathogenesis occurs when the fungi converts from its yeast form into its mold form, and this conversion acts as its virulence factor. This is commonly and most prevalently seen in premature newborns. To diagnose Candida meningitis, we isolate Candida from the patient's cerebrospinal fluid in order to confirm the presence of the fungus in the central nervous system. To treat this infection, antifungals are used, such as amphotericin B, fluconazole, and echinocandins. In order to prevent this infection, it is important that we maintain proper hygiene to prevent overgrowth in the areas where Candida is a natural inhabitant, such as the skin, oral, vaginal, and gastrointestinal biotas. It is also important that we use prescribed antifungal medications from our doctors when needed. The next form of fungal meningitis is coccidioidal meningitis. This can be caused by two different types of fungi, including Coccidioides immitis and Coccidioides posidazzi. Coccidioides is a fungi grown in the dry desert soil in the southwestern states of the United States. Currently, this species is causing an endemic in these states. The dry desert climate air causes the fungal mycelia to break into arthroconidia. These are single-celled spores that can cause valley fever, which is a type of pneumonia, when they are inhaled. These spores can remain in the air for long periods of time, and the illness can arise approximately 1 to 3 weeks after they are inhaled. Once the lungs are infected, which can be after weeks to months, the infection can spread to the brain and to the spinal cord. Fatigue and the joint pain may be the only symptoms of the infection, but they last for long periods of time and can last up to months, giving this disease its nickname desert rheumatism. Other signs and symptoms include severe worsening headaches, blurred vision, and an altered mental status. The headache is the most well-known symptom associated with this infection, so it is important to get checked out by a doctor when experiencing any of these symptoms and visiting any of these areas. Similar to the diagnostic techniques of Candida meningitis, coccidioidal meningitis is also confirmed through the isolation of coccidioides from the cerebrospinal fluid. Also, patients' recent travel history is looked into, given that this species occurs in specific areas. The treatment for this infection is also antifungals such as fluconazole and itraconazole, but unlike Candida, these are used for the rest of the patient's life. Due to this infection being incredibly lethal with a 95% mortality rate, they can die if not given treatment within 2 years. In order to prevent this infection, it is important to remain aware of the fungi living in certain environments, especially if you live in their environments. Also, to avoid inhalation exposure to the dust and dirt, wear masks when in these environments, get treated if experiencing persistent symptoms such as the severe progressing headache, and to use preventative antifungals if prescribed. The third form of fungal meningitis is cryptococcal meningoencephalitis. This infection is caused by the fungi Cryptococcus neoformans var. Neoformans. C. neoformans is a fungus found in pigeon and chicken feces which can penetrate the soil. It has a thick protective capsule which allows for the dissemination through the bloodstream to the central nervous system. This dissemination is what causes the meningoencephalitis, which is inflammation of the brain as well. The dissemination begins after the initial infection of the respiratory tract after inhalation. This infection commonly affects immunocompromised patients, specifically AIDS patients. It is an AIDS-defining infection, with more than 80% of its cases affecting AIDS patients. However, due to recent use of HIV antiviral medications, there has been a decrease in incidence because immune systems are able to recover in these patients. However, there is still a 60% mortality rate. Cryptococcal meningoencephalitis can also be caused by another variety of Cryptococcus known as Cryptococcus neoformans var. Gotti, abbreviated C. Gotti. This species is emerging in northwestern U.S. states, specifically Oregon. The fungus is spread through bird droppings and causes meningitis and pulmonary disease when inhaled, similar to C. neoformans. However, unlike C. neoformans, this fungus does not affect the immunocompromised. It affects the immunocompetent, meaning these people are healthy individuals. The diagnostic method of cryptococcal meningoencephalitis is very different from the previous two forms. It is diagnosed by using a stain
called India ink, which is added to a patient's cerebrospinal fluid. This stains the background and makes the capsules appear as halos surrounding the fungi. This prominent capsule is what confirms the diagnosis of the Cryptococcus. The treatment of this is very similar from the other fungal infections, using antifungals such as amphotericin B, fluconazole, and fluoxetine. However, these antifungals are used all at once. To prevent this infection, it is important to remain aware of the environment and where this fungal species may inhabit. It is also important to maintain HIV antiviral regimens, especially if you have AIDS or are an HIV patient.
Hello, I am Sienna and I'll be explaining 24.6 the parasitic infections and prion diseases of the central nervous system. To start off, I will discuss parasitic infections and prion diseases. Both parasitic and prion diseases mostly appear in immunocompromised individuals, as protozoa and prions can cause infection if they are able to hide from the immune system. Parasitic infections of the central nervous system are rarely seen in healthy individuals and target immunocompromised individuals. Normally, when a protozoa enters our bodies, the body is able to fight it off, but in certain conditions, especially the immunocompromised individuals, the protozoa will invade the body and cause serious disease to an individual. When talking about protozoa, it is referring to eukaryotic parasites that are made on their own and infect the body, whereas prion enter the body and change the structure of proteins which cause disease. Prions in the body are non-living infectious agents, but they are fatal as they cause disease to the brain. The prion has to deal with misfolding of proteins in the brain, but I will talk further into detail about them shortly. Shifting focus back to parasitic infections, they deal with protozoa, which are commonly found in humans. The first protozoal disease I will discuss has to do with the parasitic protozoan Toxoplasma gondii, which can infect most humans. This parasite is one of the most common in the world, and the primary host is the cat. A cat can be infected by eating contaminated meat or eating the feces of infected animals. The parasite can also be transmitted vertically from mother to offspring in human contact with cats. This parasite does not seem life-threatening or dangerous, as most infections it causes are asymptomatic, but it can be fatal when toxoplasmosis is the infection that forms. At first, the infection gives the patient respiratory issues, but then progresses to cysts forming in muscles and nervous tissue. This infection is very fatal because it can be latent for years and the patient does not know they are hosting a fatal infection. When a person is experiencing this infection, they deal with symptoms including confusion, fever, headache, retinal inflammation, and seizures. One serious condition with this infection is vertical transmission. A mother can transmit infection to a fetus, causing complications or death for the baby. There are vaccines for cats that will prevent them from getting the parasite, and there are drugs to try and treat the disease if it is caught not too late. Another protozoal disease I will talk about comes from the deep areas of Saharan Africa. The African sleeping sickness is the name for the protozoal disease Trypanosoma brucei-rhodesiense, or T. brucei-gambiense. Reservoirs for these diseases include wild animals, domestic animals, and even humans. The main reason this disease is spread throughout populations is because of the Tsetse fly. When the fly lands on an infected individual, it takes the blood meal and becomes infected. The trypanosome from the infected person is replicated in the fly and moved to the salivary glands, where a fly bites an unaffected individual. Then the individual becomes infected with the disease. The parasite infection begins in the skin and creates a painful lesion. And as time goes on, the parasite moves to lymphatic and blood circulations. When moved here, fever will appear and the lymph nodes will be swollen. As the infection gets more serious, the parasite invades the CNS, which makes it an infection of the central nervous system. Symptoms of when the parasite reaches the brain include daytime sleepiness, insomnia, tremors, and other symptoms related to affecting the brain. There are drugs developed to treat African sleeping sickness, but sometimes the individual doesn't know they're infected until it's too late. The next protozoal disease is produced by an amoeba. Naegleria fowleri is the amoeba that produces the lethal CNS infection Naegleria meningocephalitis. The parasite is found in fresh water as well as pools and sewage. When the protist gets in the body, there's a chance of a cycle developing which includes cysts, trophozoites, which is the part that infects humans, and flagellated forms. Further upon this, the trophozoites enter an individual through the olfactory nerve, following up directly to the brain. Once the parasite reaches the brain, it begins to eat the blood-brain barrier, which causes the disease mentioned. Once a patient has this infection, the symptoms begin with loss of smell and taste, and it leads to the patient hallucinating and dying just within 14 short days. The time between diagnosis and death is usually short, like other protozoal infections, because the disease progresses rapidly and is not commonly seen. There is no standard treatment for this infection, but if it is caught early, there have been trials or methods for drugs to try and resolve it. The last protozoal infection I will talk about is another amoeba-causing infection. The amoeba, Encinthamoeba, is also found worldwide in water and soil. The contaminated water or soil can enter the body by contaminated contact lenses or through breaks in the skin. The amoeba starts in the eye and can create keratitis, which is inflammation of the cornea. Then, the organism begins to integrate deeper into the eye if not treated. Once the organism goes through the optic nerve into the brain, it leads to the disease Encinthamoeba encephalitis. This organism is exposed to many people like other organisms are, but only few people contract the disease. It is more likely for immunocompromised individuals to contract this disease because the immune system isn't strong enough to fight off the organism before it gets to the brain. Symptoms are similar to Naegleria fowleri, and onset for encephalitis is slow, but usually is caught too late for treatment to work. Shifting to prions in the body, these infectious agents sometimes manage to enter the brain and interact with correctly folded proteins. After a protein interacts with normally folded protein, the protein becomes misfolded. When the proteins in the brain become misfolded, they cause disease. Prion diseases resist inactivation that would normally destroy a protein. The prion can survive extreme heat and sterilization agents that are typically used for surgery. Since the prions can resist these extreme conditions, it is worrisome for spreading the prions from one patient to another. These prion diseases are often called transmissible spongiform encephalopathies, or shortened as TSCs. They are called this because they can cause disease that spreads, make holes in the brain, and are diseases of the brain. The TSCs are characteristics in which the diseases can be grouped into. There are multiple prion diseases that can occur in humans and other animals, but they are rare infectious diseases that are usually fatal. The table shown is a list of prion diseases that we are aware of. The two most common prion diseases that we see are also shown in this chart. They are Creutzfeldt-Jakob, shortened as CJD, and bovine spongiform encephalopathy, or BSE, which is more commonly known as mad cow disease. With CJD, symptoms begin to show around age 60, and usually 90% of patients with CJD die within a year of being diagnosed. Some symptoms that are shown are failing memory, lack of coordination, behavioral changes, and other detrimental effects. BSE, or mad cow disease, is one found in cattle that has a connection to a variant of Creutzfeldt-Jakob disease, which is found in humans. Both of these diseases are fatal brain diseases, and they have long incubation periods with extensive symptoms. The numbers are low due to the fact that the diseases cannot be transmitted via respiratory tracts or social interactions with those that have the disease. The prions can be transmitted through meat that is contaminated, surgical equipment not being sterilized correctly, or transplants. Prions and safety of food or surgery is surveillance closely by the CDC because the diseases are extremely fatal.
Hi, my name is Jess Wang, and I will be summarizing the presentation as well as going over the portals of entries and exits. We've learned that infections in the central nervous system often include flu-like symptoms such as a headache, dizziness, and fatigue. These symptoms progressively become more serious over time. A viral infection that enters through the skin is encephalitis, which is categorized under primary and secondary encephalitis. Primary encephalitis, better known as rabies, may be entered through the skin. Rabies is one of the deadliest infectious diseases known to humans and endemic in a variety of animals, which include foxes, dogs, cats, and bats. Treatments for rabies is immunoglobulin and the rabies vaccine. Poliomyelitis, better known as poliovirus, is an enterovirus transmitted by the fecal-oral route. Poliovirus exits through stool or orally. Polio is very contagious and most infected patients remain asymptomatic. Treatments include the Salk and Sabin vaccines. Meningitis is a serious and harmful infection of the central nervous system. Bacterial meningitis is caused by Neisseria meningitidis in young adults, as well as Haemophilus influenzae type B found in young children, and Streptococcus pneumoniae found in infants, children, and the elderly. All three of these major bacterial species are entered and exited by the respiratory route, making the infection highly contagious. Vaccines have greatly reduced the rates of meningitis. Listeriosis, caused by Listeria monocytogenes, is a foodborne pathogen. It enters into the body through the oral and gastrointestinal tract. Listeria causes fevers and diarrhea, but for those who are pregnant and immunocompromised, complications such as meningitis may occur, therefore becoming more serious. Treatments include antibiotics and fluids to prevent dehydration. Candida albicans is the most common cause of fungal infections of the central nervous system. It is entered into the body by blood. Candida is a dysmorphic yeast that is commonly seen in premature newborns. The Coccidioides species causes coccidioidal meningitis. It is usually transmitted through the respiratory route. It affects the lungs, and after weeks and even months, it may spread to the brain and spinal cord. Symptoms include headaches, blurred vision, and an altered mental status. Coccidioides are mostly found in the southwestern states of the U.S. This infection may be avoided by wearing a mask and avoiding exposure to dust. Naegleria fowleri is an amoeba that produces a lethal infection in the central nervous system. It is transmitted through the respiratory route. Symptoms include loss of taste and smell, as well as hallucinations, which may lead to death, usually within 2 weeks. Last but not least, acanthamoeba is also an amoeba that infects the brain, found in water and soil around the world. People are usually exposed to this amoeba, but very few people are infected. The disease is more serious in immunocompromised individuals, as the organism may infect the optic nerve and into the brain. It is transmitted through the eye and the respiratory routes.